And I see that people are still joining in.
Okay, while we're waiting, because yeah, that sounds crucial. Just to mention the first item on the agenda about the ISNTD meeting. Since the last email in July, there was no movement by the ISNTD and DNDi, who were working to get a date in the diary. So it just slipped through the net. So I just wrote to them and said, could we maybe try and agree on a day in November or something, which is convenient to ISNTD and to DNDi, and then we can start sorting out an agenda for it, a schedule for it. So I've just chivvied them again. Essentially, it was on them to agree a convenient date. That's all. So I've just pestered them about that.
That sounds good.
Yeah. Well, I mean, it was the idea. It's just that over the summer, I think the email must have just got buried or something. So I will inform. I will update at the next meeting what I hear.
Yeah, the ASTMH is completely virtual this year. And there will be a session on mycetoma, including some drug discovery, I guess.
Really?
I saw on the schedule that Ed Zelstra is talking about drugs for eumycetoma. Guadalupe Chavez is talking about drugs for actinomycetoma. And I will be talking about melanin, what happens when you inhibit melanin to the treatment. There's one other talk in that session, and that's about diagnostics, but maybe for those attending ASTMH virtually, that will be an interesting session to look for.
Is Ed going to be talking about the clinical trial?
No, I don't think so, because officially the clinical trial still is not ended. Although the last patient entered and finished. But as far as I know, because I'm still blinded, I assume that everybody else is. But
okay, I'm interested to know what he's talking about, if it's about drug discovery.
So I will check where Ma went to, and then
I could then just fill in also about the issue to do with schools participating in the synthetic work while we're waiting.
Yes.
Because the stuff that Kimberly was talking about last time about the successful synthesis of the compounds by Sydney Uni students was really interesting. And the school that we've been working with here, Sevenoaks School, want to work on the project again this year. They did, I think, find the phenaramol chemistry a little bit challenging. They had a go, but it was a bit challenging. So it seemed to me that maybe we could encourage them over to the other series, because the chemistry sounds like maybe it's a bit more tractable for a simple high school lab. If that's the case, and if we have, which I'm assuming we do, just written protocols for how to do things, then all we need to do is maybe suggest a molecule that they can have a go at, which is likely to be synthesizable by a group of them with teachers. Does that sound like an okay way forward?
Yes, for me it does.
We've also been talking with John from MISA in the US about doing synthesis with his students on the 2-aminothiazoles as well.
Yeah,
so
right.
We shared with him the procedures and everything, so it's going to be easy enough for us to quickly just share that with Sevenoaks.
Right, great. Would you be able to post that to an issue on GitHub? Because I'm trying to encourage the school to join us there.
Yeah, I've already got it on there. I'll just share the issue with you.
Right.
Just another one on that. So Sevenoaks, Gabby from Sevenoaks wrote to the Breaking Good email address and asked if they could share a video that they made last year on the Breaking Good site from OS mycetoma.
Wow.
And they've sent that across. And then I have replied to Gabby to say, does she want us to write something up on GitHub, like for OS Mycetoma or for on one of the websites, or on Breaking Good website to go with the video to explain what they're doing, to sort of have a bit more of a case study so that we can start populating with some schools.
Right.
But of course, we want that to be cross-pollinating a different website, so it can go in I think we can have the same video and even piece that's shared across different platforms, if that's something you'd be happy with too.
Yeah, great.
And I think there are also some schools in Sydney who'd likely want to start that next year. And who else? There's somebody else. Oh, Washington. I think Washington Tacoma may want to do something with that. Hey, Kim, if they're back in the lab next year,
I think so
they've
as
been
well.
really engaged with the essential medicines and synthetic route design towards overpriced medicines. Some of them are really good, the work that they've done there.
Right. Okay, that sounds really good. I guess if we're going to have a situation of multiple people maybe making molecules, then we just need to clarify targets, and which might be useful and non-duplicative. I think that Seven Oaks would like me to do the sort of little intro talk again about what the whole thing's about, which I'm happy to do. And it would be cool to show them targets. If you, in the course of doing what you're doing, if you happen to have molecules that need making or could be made with commercial materials, that's great. If you could share those, that'd be great. If you don't, then maybe we should come up with a few as potentials,
Yeah, that sounds good. And maybe it's a good chance for them to even try using GitHub as the lab notebook, which is something Kim has been working on together with Clementine, of trying to experiment with whether having everything in one place is a solution, and also something that people can sign up to learn how to use one platform rather than moving between different places, if that makes sense. So that could be a possibility too, Kim, probably with some of the things you're writing at the moment about GitHub as a lab book.
Yeah. And I think that's kind of the next step. I think Clem and I have used it for our organic synthesis, and it seemed like it's okay for making molecules and characterizations of them. The only thing is ChemDraw, we use ChemDraw right to and then we just insert an image of the actual reaction. So whether the students have access to that or whether we direct them to a free open source alternative that they can use, that would probably be the main consideration. I think the rest of it's okay in terms of actually using it. And we have templates as well that we use for our experiment. So we'd probably be able to share them with them as well. So there's some consistency between, which will be beneficial for when we write up and stuff.
Right.
And we have plenty of the open source drawing tools, but I think there's also something particularly for schools that can be quite beneficial in them drawing the molecules and even taking photos to upload them, because I think that's almost something, I don't know how others feel, but I felt like drawing reactions or drawing molecules was critical to my understanding of chemistry, and sometimes we're almost skipping that by asking people to draw things using templates online. I think you can sort of miss something there.
Yeah, I can definitely see the value in actually drawing it by hand versus only doing it online, especially in the learning process.
Yeah, for the series 2 molecules, so the aminothiazoles, the ones which were made, the original molecule that had activity in vitro, so the one which was remade by Kimberly. Unfortunately, the ones which were made by the students, they didn't have activity in vitro, so there needs to be a little bit different scaffold than, side change than the ones made previously, I guess.
Yeah, so I think there's a key in that the section that we changed from the original one, I think it was a 2-pyridine ring. So we've identified that moving to the 3 or the 4 position with that nitrogen reduces activity. And also we've then looked at substitution at the 2, 3, and 4 position of that ring. So I think at the current moment, keeping that pyridine ring is advantageous, and we might look at the ring at the other end of the molecule and start maybe explore a little bit in there and see if it accepts some modifications, because at the moment from what we've done it sort of looks like maybe that's the pyridine ring might be important, and it's possible that also we see that in the pheneromoles as well, and maybe that pyridine ring's binding at the same site, and then it's just playing around with the rest of it to get better. I don't know about that though, that's very like fluffy, but like we also saw that pyridine rings were important in the pheneromoles series, so maybe that's something we want to keep. But yeah, the synthesis route would work fine as well for doing modifications to the other ring system. So we did it so that the students did the same first 2 reactions, and then they just modified the 3rd one by adding in a different substrate. If we wanted to modify the other one, it would just be the opposite way around. So in the first step, they would each have a different substrate, but do the same reaction and then carry through the next 2 same reactions. And we should be able to just do that. So it should also be quite simple to look at modifications of the other ring system.
Okay.
Kimberly, just a quick question. I'm doing writing minutes for GitHub. Can you please repeat some information regarding the what you said about series 2? So which region would you like to explore again? Can you just quickly repeat that?
Yeah, so there's 2 ring systems. There's the ring system that actually comes off the amine, and then there's one that's not off the amine, basically. I think that's the easiest way to describe it. And the one that we've played around with at the moment is the one that comes off the thiazole ring. And what I'm talking about here is to start exploring the ring system that is attached to the amine.
Yeah.
Does that make sense?
Yeah.
Yeah.
Okay. Other points on this this topic we need to discuss?
Yeah, I just wondered whether there was an up-to-date SAR diagram for anything we've learned about the activity of these molecules to date, or is that what we're going to talk about now?
Well, maybe it's best to share first the in vitro activity which we noted. We uploaded it also on GitHub in the table. And then maybe from there we take the discussion to the synthesis and complete the round with the SAR, or the other way around. So shall we? Okay. Yeah, shall we present a biological evaluation? And then I will share my screen and Mark can present our results. Let's see if it's... Can you see the screen now?
Yep.
Yeah. Okay, go ahead, Mark.
Yeah. Hello, everyone. Today, the presentation is about the biological evaluation of MycetOS Open Source compounds. Yeah, the compounds we received in total 36 compounds from the Kimberly and the Mark, I think. I forget that. Yeah, of this 36 compounds, and 22 belongs to the series 1, and 12 compounds belong to the series 2, and the 2 compounds belong to the series 6. Yeah, next to the 36 compounds received, has analyzed indicated several compounds to be analyzed in vivo, and I checked those compounds data in the master Excel, and we found the just one compounds. The first one, MycetoS 00014, its MIC is below 8. So we just so we know how only a compound not able to inhibit growth at 25 millimolar micromolar will not be tested in the larvae model because the concentration we use for the treatment is only 20 micromolar. And we started the Vitro test about the 36 compounds. And firstly, we did the growth inhibition with 36 compounds and at 100 micromolar and 25 micromolar. And we can find the 15. We selected 15 compounds to do IC50. And we found the MIC below 8. We just got 5 compounds. Next. The results show in the table just like this, and we can see that the compounds inhibit the fungal growth at 25 micromolar. We can find just one compound from series 6 and one compound from series 2, and others compounds from series 1. And we can find the MIC median below 8. We just selected 5 compounds. The first one, the DM27-1 and DM34-1 and the DM37-1 and MycetoS 00166 and the MycetoS 00174. But the next, the MycetoS 00175, the compounds is the compounds inhibitor the growth, the fungus growth at 25 micromolar. But I checked my result. When I checked my results, I just forgot that. So I missed this one. This compound.
So this will be added.
This will be added. And the last one, the compounds called MycetoS 00176 from series 2. It works well in it already tested in our master Excel and it works bad. So it works well. So I didn't select it. And added the compounds from herbs. The first one, EPL-BS0115. The MIC is below 8. So added those 5 compounds we selected. In total, we had 6 compounds to do in vivo test. This is a vivo testers survival curves. And we can see the circle. The circle line is adequate. We can find they are almost 100% 100 survival, and we can see the.
So these are non-infected larvae treated with simple aquadest. This demonstrates that all the larvae were healthy. Continue.
And we can see the infected control. In the 10th day, almost all larvae are almost all larvae died in 10th day, and the others group is treated with treated with those compounds, and we can see the triangle line is stand for the larvae treated with amphotericin B. We know the amphotericin B works well in larvae model, so we selected this one as our positive control. We can see the amphotericin B treatment group has the significance with the infected control. And as for the blue one, the blue one is MycetoS 00166. Yeah, it works well, and the p-value of this group compared to the infected control is below 1.1 no 0.0001. So the MycetoS is a promising compound we want to select it. Yeah, and for the as for the MycetoS 00166 and the EPL BS0115, the two compounds we finished 3 times test and 45 LV per line. But the EPL BS0115, the purple line, it's just like the infected control. So there are no significance with infected control. About the red line, the MycetoS 00147, it may be prolonged larvae survival because now we just have 5 larvae data of this. So maybe more data need to be added, and we can find the final results. And the green one, treatment larvae died in the in the 3rd day. So maybe it doesn't work. And other than MycetoS 00166 and EPL BS0115, other compounds still need more tester. Still need more tester in the future.
Yeah, so what Ma meant is that MycetoS 00166 and EPL we tested 3 times, so those experiments are finished. The other lines we still need to repeat to get enough power, because these consist only of 15 larvae, and that's not enough power to get something statistically significant.
What is 166?
Yeah, 00166.
What is it?
It's
a Fennery mole.
Fennery mole.
So it's HPD 0451P3 Christ. But so probably
yours, right?
Hardy knows better what it is than we do.
I might have to dig up the structures. But
because that looks like the best in vivo figure we've ever seen.
Yes, it does.
So I'm interested to know what it is. And also, just to go back one slide, sorry. Sorry, go back again. So the MIC here of 0.25, which looks very nice, that's 193. If you go forward, next slide, sorry.
Yeah, that one is one of the compounds still need to be tested because we had to do it batch-wise.
Right.
So those are still up and running currently.
That's an oxime. That's the perfinox oxime.
Right. It'd be interesting to see how that goes in vivo.
Yeah. So that's the series 6, and due to the sake of time, we decided to go first for the Fennery moles to finalize this data set, and then add the series 6 compounds to the last test we're doing.
Wendy, I have a question. What about MycetoS 191? Does it still need to be retested again? Because
yes, it needs to be tested again. I don't think it is here already, but I don't think that the line was there. It's in the file to be filled in, but there's no data yet. So it shows the line already, or at least the legend, but the line itself is not there yet. So yeah, in the near future, we simply add the data.
Yeah. As for the MycetoS 00193 from series 6, still need to be tested in vivo. And I missed the one, and the one I missed. Yeah, due to sake of time, we selected the pheneromous compounds first. Yeah, we already prepared new fungi to test those compounds. We need to repeat. Yeah, this Lavi season will end. I'm not sure we can finish them all this summer. So yeah, but we could finish the pheneromous paper this year, I think. We will make figures when all survival data is known. We will update the figures on the log-D act when the survival percentage are unknown.
Yeah, so the only thing, because I put the data in yesterday in the sheet online, I noted that the log-D of the new molecules is not there yet. So we probably need to ask Ben to calculate those as well. So all the other data is not there yet.
Yeah, and another thing is, I think one or two of the compounds that Ben tried, I think one of them missed a log-D as well. I flagged that last time, but I don't think he fixed that yet. So maybe when he's doing this, he can add that one in as well. This one that just missed a log-D.
Yes.
Yes, Kimberly, I am happy to do that as well. So that I'll show you in a minute what that 45-1 crystal looks like as well. But yeah, I haven't added the latest 36 compounds data into the SAR just yet, but I can do that very, very quickly based on the current structure. But I guess the next steps in terms of like just getting the SAR going as well, just to select the most relevant data for our paper. So I guess the way that I did it is I looked through all of them and I analyzed all of them, but I guess some, due to wordings or, restriction, and we might need to just shorten that down a little bit. Yeah.
Okay, shall I stop sharing the screen? Or are there any questions still related to this?
Oh, sorry. The shipment that Epichem sent to you, were you able to do? Was that data in here as well, like the in vivo of interest?
Yes. So the EPL BS 0115, that was one of the compounds in their shipment. But we also looked at the other compounds, but they were all with an MIC. Well, they were not inhibited 25 micromolar. So since we only treated larvae with 20 micromolar, it didn't seem very relevant or feasible to look at the in vivo activity there. Because yeah, if it's not inhibited in vitro, it will not be inhibited in vivo. But we decided to take EPL BS 0115 as one of the first molecules. But unfortunately, it was acting exactly the same as the control line.
Yeah.
So it didn't seem that that compound was prolonging larval survival.
I guess that's interesting as well, because the reason that I suggested those was because they had really good in vitro the last time, but I guess it just, for one reason or another, it didn't quite, it's not repeatable.
Yeah, so probably you did the SAR with the 100 micromolar.
Yeah.
So this compound did inhibit at 100 micromolar,
okay, yeah.
but not at 25. So maybe if we repeat the SAR at 25 micromolar instead of 100,
okay,
the results will be a little bit different.
Yep.
Because this one wouldn't pop, well, BS 0115 would pop up, but the other not, because they were not inhibited at that concentration.
Okay.
So the MycetoS 00174, That basically kills all of them, right? Is that what this graph showing? Because the interesting thing is that's very similar in structure to the 166.
Yeah, so this is only one experiment, so it is currently repeated.
Yeah.
But from this one experiment, it looks like it might be toxic. We still, because we did a quick and dirty, because when we were able to start, it was already nearing the end of the larvae season. So what we're currently also doing is doing the tox data in the larvae. Normally we do that simultaneously. So in this last experiment, the toxicity will be taken as well. So it could be either that the compound itself is toxic to the larvae as well. That would then make a reason why this one dies so quickly. The other reason could be that the simple adjustment between 106 and 174 makes the fungus maybe more pathogenic.
Mm-hmm.
Is there a version? Is there a compound? Sorry, I can't remember. So the active compound 166. Imagine that one, but with a fluorine. You know, sort of an analog, basically the same version as 174, but with the bromine and chlorine switched. It would be interesting to compare the activity for those two compounds.
So you're saying like the 166 with a 2-fluoro?
Yes.
2-fluoro for bromo?
Yeah.
I think the one that you showed there was not the right one. I'll have to double check, but I think what I have on file was a bromo version is 166.
Yeah. So the bromo version is 166. The 174 is a 4-fluoro 2-fluoro version.
Yeah.
So Alice is wondering what happens if we have the 2-fluoro on the bromo one. What modification, like what difference in activity do we have there? I think it might be in some of your SAR. I think we talked about this.
Yes.
Yeah, I think we have to look back because
it
is really interesting. I think it'll be interesting, you know, and with the repeat experiment and then having a look at Dimitri's compound. I think Matt's sort of written something about that as well in the notes to compare those, but it's pretty exciting.
Yeah, we were very glad to see this activity because last year we had seen so little activity amongst the Fennery moles that we were quite surprised that this one worked so well, because it's even working better than amphotericin B, which was so far our best molecule.
That's really cool. So in terms of taking these compounds and this data, what do we think are the next steps in terms of progressing towards the paper? Wendy and everybody, I'm just going to grab my dog. Sorry, I'm going to switch the video off for a second.
Yeah, I think that's first we need to finish, of course, this survival data. Then I will make the comparison which I made with the log-D, etc. Just simply add the new data and see what comes out of that, because we weren't able to do that yet, because first we only finished 00166 and the EPL compound. And we need the log-D for that. So I only noted yesterday that the log-D was still missing. So I will send Ben a message to ask if he could calculate that. From the results from last year, the correlation still stood. So hopefully this will still will improve the correlation, or yeah, at least it will make it stronger, I hope. I also don't know if, but yeah, since the log D is not available, if this one has a very low log D or if it has a high log D.
Yeah, that would be interesting to know, because if it's the opposite of what we want and it's our best compound,
then our hypothesis should be
throws a spanner in the works, doesn't it?
I can show you a table that I did on the SAR, just like just an overview of like log D versus activity as well.
Shall I stop sharing? Then you can share.
Yeah, sure. So this one is again on 100 micromoles. I'll see if I can do one for 25 micromoles in like after, but this one's at 100 millimoles and it doesn't have the 36 new compounds. But here's like an overview of that. So what it is I just split them into like the less than 20% growth. So firstly, sorry, there's 5 different motifs. So all of the compounds that we tested, so if I accept the 36 new compounds, how it all fall into these 4 different structure here, or so like simplified structures. So like the first one is triarrow, then you got the piperazole, which is this ring here, and then the piperazole, which is these 2 guys here. So these are at 100 micromolars. There's kind of 3 different groups. So the group that's of interest is the one that's less than 20% growth, which is in red.
You want to share? You want to share your screen?
Oh,
do you want to share your screen?
Oh, sorry, I misshare it. I thought I shared it, but anyway, I would do that.
Yeah, I was looking for it if I was on a different view, but
is that on now? Sorry.
Yes, I can see
clicked share, but for some reason it didn't. But okay, everyone can see it. Okay, so this is the overview of the SAR that I did. So there's 4 simplified structures that I kind of like deduced out from all of the compounds so far, except the 36 new compounds. So it's either like in this structure, it was like a triarrow, so 3 aromatic rings, or a piperazole, which is this ring, and then a piperazole, which is this kind of structure here. Growth is at 100 micromolars, and I split them into 3 groups. The one of these of interest is the one that's less than 20% growth at 100 micromolars, which is I put in red here, and then the other ones are sort of like, it's still interesting because you can see kind of like what works and what doesn't work. So in this case, these 2 guys will kind of like give information about what's not working. So for the first 2 general structures, the triarrow and the diarrow piperazole are the one that is most promising. Motif B, we've done a lot of tests on, so 40 unique structures. So some structures have, we also do repeats as well, or retesting, but these are 40 unique structures, 25 for this group here, and you can see a 68% of the compounds in this group here has a complete inhibition at 100 micromolars, and then about 40% is in these structures, and then these ones are a little bit lower. And then of these tested compounds, they're all roughly so like 50-50 in terms of like the log D values at pH 7.4, so log D less than 2.5 and log D more than 2.5. So we did have a diversity of log D as well. And of the compounds that exhibit complete growth inhibition in vitro at 100 micromolars, then these first 2 again have fairly sort of like 50-50 to 60-40 ratio of log D less than 2.5, but log D more than 2.5, and these ones sort of like have the opposite. So they have more log D is larger than 2.5 versus less than 2.5. But yes, this is like an overview of all of the compounds that I've done SAR on so far. at 100 micromolars.
Yeah, what we noted that as well is that in vitro and in vivo look different. So
yeah,
in vitro we saw more inhibition with higher log-Ds in general. So maybe the criteria for a molecule in vitro and in vivo are different.
Yeah. So I guess that most of the that is true from here as well. So most of the compounds here do have the in vitro that is larger than 2.5. So I guess it does show here as well. But yeah, so that, yeah, this is just what I quickly put together. But yeah.
Yeah, but interesting that you see that certain side chains favor activity over others.
Yeah, so some of the some of the motifs or some of the general structures do perform better than others. So like the simplistic one with the 3 aromatic rings generally do perform the best. And then the more you kind of tweak and tinker with them, so adding extra length or adding extra side chains, it does actually have an impact on the values, on the in vitro and in vivo value as well. So yeah, that's what I've done. But I guess the next thing that we can do is just, as Matt put in just shortly before he left, is just focus on around the potent compounds, which what I kind of did, but just need to shorten that down into a more succinct or concise, and then you might need to remove some of the less interesting one, or just talk about it in a more generic term as well, just to shorten it out. Because right now I think it's about 5,000 words or 6,000 words, and it takes multiple pages, so it does need to be shortened down.
Yeah,
I think we can trim that right down for the paper, but because we, the diagram will help, and probably feeding in some of Dimitri's responses too.
Yeah, and this one is specifically only on series 1 as well. So I haven't actually included any SAR for the other series as well. So that's the other thing that this SAR lacks.
yeah, but would sorry, maybe it's a stupid question, but since we assume that the other compounds might have different modes of actions, would then that always be true? So because for a compound to work, it needs to penetrate, of course, in the grain. So those features should be the same for all molecules, probably what is needed, but it should also fit in within the target. And I can imagine that would be different if the target is different, because the phenol it's supposed to be the CYP51, but maybe the aminothiazoles or the series 6 have a different target within the cell. So then that would also mean that there might be different characteristics needed to interact with the target.
Yeah, I see what you mean. Yeah.
The azoles, though, they have the same targets as the phenol, so maybe that will be applicable. But I'm not so sure. I'm not sure about series 2 and series 6, what their mode of action would be.
Would it be helpful to look, for example, for series 6 at the mode of action for the perfinox positive control? Because it's like a non-fungicide, or it does not help much. I mean, like in terms of information.
Yeah, if it's quite similar, then probably that's also why we, because we never proved, of course, that for the phenol the CYP51 in Madurella is indeed the target. Because it was known to be a target for Chagas disease. So if this is a similar molecule as a target in a different fungus, then it's most likely that Madurella would have the same target.
Yeah, and one thing I note about the SAR as well is I read through the papers that Keenan did for Chagas disease as well, and there are some functionalities or some of the motifs that do have similar activities as well. So like when you switch one thing to another, it goes from high activity to low activities for both the Chagas targets and also for mycetoma. So I think that it does provide evidence or enhance the chance that it is the same target as well between these two pathogens.
Yes. So are there updates? Are there other updates you would like to share?
Yeah, I would like to update you on ECL work.
Okay, go ahead.
Can you see my screen now?
Yes.
So I would like to start off with the piperidine motif exploration. So from UCL, we have currently at UCL, I have made 5 compounds in total with the latest edition DM40/1, which complies with lower than 2.5 log D value preference that we set out previously. And this adds in total to 5 compounds with the piperidine, because previously we found that DM7 promotes almost 24% larval survival. So this is something that we would like to explore further. With respect to the biological assay results, we thought that it would be useful to have a look at the in vitro and in vivo results, and then rank those molecules with the piperidine motif based on their potency. And after that, maybe identify and rank them and identify the most potent piperidine containing phenomenon, and then work with the motif by doing attachments at different positions. So say, for example, 40/1 is the most potent out of all piperidine phenomenon. So we would do a metapar ortho positions and so on. We thought that might be useful. Also, what we've been doing recently is we came back to piperidine motif exploration, and I made some compounds from the master list, which are fairly lipophilic, maybe in some cases slightly above what we would usually want currently, but those have been made and currently undergoing different stages of characterization and purification as well. So this is just the summary of synthesis that has been done. So mainly I opted for Buchwald-Hartwig amination, with exception to bromocyclopentane, of course. And recently I tried microwave synthesis as well, which worked perfectly well with the latest edition. So all those compounds have been synthesized already. Some good news came from John from IMCA. So they made those 3 compounds and sent them to us. We received them and confirmed their identity. So we have 3 additional compounds ready to be evaluated in the future biological assays. And those are in the master list as well. Also, John asked us to send him an SDF file of the master list compounds. So I guess that comes back to our conversation at the beginning of our session, whether the schools will want to carry on. So apparently he's looking at more options from the master list as well. So we'll be in contact with him about this in the future. In consultations, I think we definitely need to probably help them with targets and maybe give some priorities as well. Yeah, this is just a summary of what's already completed. Oh, sorry, I missed one. I missed the piperidine with the 4-hydroxy motif in the scheme. So we have 7 compounds ready for the next round of evaluation at the moment. Just to finalize, a few words on the metabolic clearance. So we've been talking about this on during our previous meeting. So I collated all the EpiChem data available up to date and generated this metabolic clearance versus log D. And that's how it currently looks. But I think what I should do is probably get a modified version of this because we need to zoom in, like say from 2 to 5, just to see like a better pattern. But this is just like a general overview, summary of what's the metabolic clearance pattern is, how it looks like. Yeah, and those are the compounds that give the highest values. So I would guess they're the least preferable ones. So this is just like a summary as well.
Do you know if any of these compounds were tested? And because they also have a different code again,
so those codes refer to the EpiChem library, as far as I can recall.
Yes, I know, but do you know if some of these were in our own list and tested?
I need to double check this. I'm not sure. I don't think so.
It doesn't look like.
It doesn't look like it. I think those were synthesized by EpiChem previously. Tested.
Okay.
But I will double check for just to be absolutely sure.
Because we did test a few of the EpiChem compounds, and so I assume that you know the clearance rate of those.
So it should be, there should be some data on that as well.
Yeah, so maybe that's good to include as well.
Mm-hmm.
Because the most ideal drug would of course be one which can reach the target for a long time. So if it's eliminated very fast, then it still might not be the most ideal one.
Yeah. So we need to check whether there were any compounds tested by Erasmus from the list.
Yeah, it would be nice to know what the clearance rate is of the molecules we have, and if they're, of course, we don't know of all the compounds you made yourselves, but maybe there is a correlation with those few for which we do know. And of course, a clearance rate in a mammalian model is different than that in a larvae model, but still.
Okay, then I'll give you an update for next time we meet, definitely. But actually, earlier than that, I'll just post an issue on GitHub.
Okay.
Under the biological evaluation issue, probably. Yeah, I'll do an update on this.
Any other updates or questions?
So I'm done with the presentation. That's it.
Okay, yeah, for me it's very interesting. You see some outliers which have a very high number and some which are really at the bottom. So yeah, it's very interesting that there's so any difference.
Yeah, I've talked previously with Matt. He said like it would be better like if we would look more closely at the say from 2 to 5 log D value range, because it's a bit like overlapping too much, so it's hard like to see. So we're definitely getting a modified version of this as well, like a zoom-in version.
Okay.
Yeah, but that scheme contains all the molecules with the highest clearance values. So I did this on purpose.
Okay.
So that would be helpful. I think it's on GitHub already. I posted this previously, like a long time ago.
So is the criteria for the best one in here would be highest clearance, or how do we look for this?
So if the values are too high, then it means it's getting eliminated like very fast, which is unfavorable.
Okay.
So those that we see now are not the priority, I would say. They're like the worst.
Okay. So do we want lowest clearance, or do we want kind of like in the middle of it?
So if you get a lower metabolic clearance, then you get the drug penetration potentially, and the drug actually in the organism for a longer period of time. If you get like something like this, then it means like it gets eliminated like very quickly. So it might not just undergoes metabolism like too quickly and might not exert like any pharmacological action or chemical.
Yeah, so what the exact, the most ideal metabolic clearance would be, that also depends, of course, on toxicity. So if a compound is very toxic or accumulates, then you want a little bit higher metabolic clearance rate. If it's if it's non-toxic, then a very low clearance rate would also be very good. So it's a total picture, of course.
Yeah, but I think it would be useful. I think that's the reason why Matt emphasized the fact that we need to look more carefully at this region, because it's overlapping, and that's the most useful information, basically.
Yes.
Well, everything is useful, but like we need to see maybe, because we were kind of like looking for log D values preference less than 2.5. So at least that's what we're looking for, like when synthesizing compounds as a preference. So I think it would be useful to see what's the correlation here as well. Yeah.
Okay. Are there any other updates we need to discuss? Maybe Dimitri, can you stop sharing your screen?
Yeah. Oh, just one last thing. Would it be possible to get all the data on those compounds in vitro that have been tested recently?
Yeah, the in vitro data I uploaded yesterday in the. But I'm not sure if DM40 was there.
No, DM40 is the new one. Is the new one.
DM40 is already updated.
Yes, so that one was inhibited at 25 micromolar. I don't have the full data set here on the screen, but what we have, we uploaded.
Okay, we'll have a look at this. Yes, we'll probably explore more piperidine, but like with different positions. So as I said, like we will rank them based on the preference, like based on the potency, sorry, and then maybe have a look at different substituent position in the same compound.
Yeah, I just had a look, Dimitri. All of the compounds on this screen, they're in vitro are all up on GitHub in the master list.
Are you looking at the Mycetoma Excel file now currently?
Yes, the master list. These one you're showing right now, they already have in vitro.
41 is a new one. It cannot have any data. It has been synthesized.
40 is already on there as well. It's the very last entry. Oh, Oh yeah, I missed
Oh,
that.
it's just like
Okay,
I
okay,
add
sorry.
all new entries as I go, just to make sure that everything
It's up
is
to 39. It's up to DM 39. So yeah, sorry, 40 is not there. I missed that.
It's a new one, so
Yeah.
it will be in the next biology class. I also noticed an interesting information based on, oh, sorry. So for example, this one on the upper right-hand side is an oxime. So like in terms of structural consideration, that would be useful as well, because it gives us information that might be, that oxime's potential might have like high metabolic clearance as well. Because for example, the peripherox, which is a positive control, almost has the same structure, but the only difference is 2,4-chloro position in peripherox. So that was kind of useful as well.
Since we're here, I can tell you something about this one as well. So the second one from the top left, so the one with with amine core, that one will generally have poor in vitro. So the core amine is not a desirable in vitro one. And the pyrimidine, so the two on the second row, the two on the right, those ones you would expect them to be performing poorly in vitro. So in the pyrazine, which is one down in the bottom right corner, pyrazine ring is also kind of like not good in vitro. So most of these you would expect them to perform poorly as well, from an inhibition of in vitro point of view.
Even with the piperazine motive.
The problem is the pyrimidine, so the two nitrogen and the aromatic ring on top. So that will be So if it has a pyrimidine instead of a pyridine, usually you would expect a poor in vitro. So anything that's not pyridine on the top ring will be quite bad. So things like pyrazine, so like the two nitrogen opposite each other, or the pyrimidine, which is the two one on the right. So yeah.
Sure. Yeah, I was just slightly confused because like we've been working on the piperazine mode of exploration. So we made like those 3 new compounds. And John actually sent similar ones as well. Yes. Yeah, kind of similar ones. So the next batch will be like largely, well, at least at the moment, it's largely the piperazine mode of exploration with the 4-ring attachment. But we'll need to see what the results are and then come up with conclusions. Yep.
And so if you have those molecules already, although the larvae season ends, that doesn't mean that we can't do in vitro analysis, of course. In vitro is independent of the larvae season, so we can do that in the winter as well.
Would you prefer to have it like as soon as possible, or shall we make some more and then send it to you, like in a month's time or like in 2 months' time?
Yeah, something like that. A little bit earlier than the start of the larvae season is preferable, because then we can start already early in the larvae season if we need.
And the next larvae season is
starting in April.
April. Okay. Yeah, that's not going to be a problem. So I would expect to have another parcel sent like before the end of this year for sure. It's just a question like whether we can send like in several batches or just accumulate like more analogs and then dispatch them to you. But we'll see. Yeah. But at least at the moment we have those 7 already. Yeah.
Yeah. Okay.
Okay. Thank you.
Since we already passed the hour, are there any other things we need to discuss, or shall we continue next month?
That's all for me. That's all for me.
Okay, then. Yeah, it was nice seeing you all after the summer break. And hopefully next month we will have all the in vivo data we need. And then I'll see you all in October.
Yes.
Bye, everyone.
Bye.
Bye.
Bye, everyone.
See you next month. Bye.